Hi everyone, and welcome to our video on the factors affecting the evolution of a species. This is a video in our Module 6 Part 2 series looking at the patterns of inheritance, and what we'll be doing in this video is have a look at specification reference 6.1.2e, the factors that can affect the evolution of a species. And what we'll be doing is having a look at these two kind of key types of selection, stabilizing and directional selection, before we then go on to have a look at genetic drift, genetic bottlenecks, and the founder effect. So when we're talking about evolution, then we need to talk about natural selection. The two go hand in hand, as Charles Darwin told us many years ago. Now, what we actually need to think about before we delve into natural selection is how new alleles can actually arise within a given population. And there are two key ways. First is mutation, and remember, mutations are randomly occurring events. And the second is migration. So if we're talking about organisms whereby a new organism enters a particular population, they could very well bring with them some new alleles that were not present in that particular area at that time. So hopefully from GCSE we all remember the basics of natural selection, that when we are looking at a population, there is variation within a population as a result of these random mutations that have occurred over time. And that means that some of the individuals within a population will be better adapted to the environment in which they live. Those ones that are better adapted, so they've got these advantageous alleles, will survive and reproduce, and that means those advantageous alleles will then be passed on to their offspring. And what we see is as that repeats over many generations, then the allele frequencies will change and they'll actually start to favour those advantageous alleles. They become more prevalent within the population, and those that are not advantageous, that may even be disadvantageous, they're going to decrease in frequency. So what we see is as a result of this natural selection that is occurring, the allele frequencies within any given population will change. Now, the first type of selection we're going to have a look at is what we call stabilizing selection. Now, we will see this stabilizing selection if we've got an environment that is remaining unchanged for a prolonged period of time. Because if the environment is remaining unchanged, then what we are going to see is there's nothing that's going to be selecting for different alleles. So instead, what we're going to find happening here is we're going to be selecting for intermediates, because we don't really need those extreme scenarios to be catered for, because the environment is pretty constant. So where we're not seeing those dramatic shifts in environmental conditions, we don't really need those alleles that cater for the extremes. And as a result, we are going to see that favouring of the intermediate, the kind of happy medium, if you like, phenotype. And what we're going to see as a result of that, we can actually reduce the genetic variation in a population, because those alleles for the extremes, if you like, are going to be selected against over time. And a good example of this is actually in human babies. So when we consider the mean birth mass of human babies, it is about 3.5 kilograms. So that's your kind of mean baby birth mass. And the reason that that is the mean baby birth mass, it's because that's where they're most likely to survive. Now, obviously, as we've already said, if an organism is more likely to survive, it's more likely to then go on to reproduce and therefore pass on its own alleles. So what we have seen here is a great example of this stabilizing selection, and another really good example of a type of graph you could be asked to interpret on your A-level. So what we can see in the graph that I've given you at the bottom here is we've obviously got three different axes labelled. So on the x-axis at the bottom, we've got our birth mass in kilograms, and then we have two different y-axes. This is where you need to be careful and think in your exam, okay? So on the left-hand side, we've got the percentage of the population, and on the right-hand side, we've got percentage mortality. And then do notice that at the bottom we actually have a key for our two different lines. They're different colours here. So we've got the blue one, which is the kind of dome-shaped one in the middle, and then we've got the red one, which is the U-shaped one that we have obviously on top of it basically. So the blue one, the population, the red one is our mortality. So if you were obviously interpreting this graph, you would have to make sure that you use the key to know what we're looking at. So if we're reading mortality, for example, then we need to look at our red line, and then we want to be looking obviously at the correct scale at that time. So just go careful. So what does this graph actually show us? Well, as we said, our mean baby birth mass is about 3.5 kilos. Now on this graph it's a little bit less than that, because if we actually track down, it is closer to the 3 on this one, but you get the idea, it's around the 3.5. So what we can see there, that is the majority of births come in around that point. Okay, so we can see you've got a real peak there. And the reason for that is that our babies are going to survive. Have a look at the red line. The percentage mortality around those little babies is only at about 2%. So that's obviously going to be most of them surviving, and therefore they will pass on their alleles. It then drops off really quite quickly. If you have a look at the number of babies, so the percentage of the population that have a mean birth mass at those extremes, either the high or the low, then it really is very low at those points. And the reason for that, have a look at our red line again. If we look at the percentage mortality rate there, then what we can see, if we come across, let's just take this little point here. Okay, so we're going to come in round about, what's that, about 1.3ish, I'd guess. So about 1.3 kilos with this very poor scale. You get a better one on your exam papers. And then as we come up, we can then come across, and what we find is it's about a 70% mortality. So those babies with that mean birth mass of roughly 1.3 kilos, about 70% of them are going to die, at least when this data was put together. The good news is that this is actually quite old data, and therefore we have improved our techniques for keeping little tiny babies alive. And in fact, you can see my little tiny baby in that bottom corner. So that's actually my son, and he was born at a grand total of 1.23 kilograms. An absolute whopper. Not really, he was tidgy. As you can see, that's my hand there to give you an idea of how teeny tiny he really was. But thanks to the amazing staff at the William Harvey Hospital, then he is a thriving, bouncing 4-year-old these days. And even though obviously we can look at our graph here and see how the odds were kind of stacked against him, it goes to show how obviously healthcare has improved, and therefore our little extremes do go on. And believe me, he wasn't the smallest baby in the NICU at the time. And I believe I'm right in saying, at this point in time anyway, that the tiniest baby that has survived, at least in the UK, is only around about 300 grams at birth. And that is absolutely crazy, if you ask me, that we can keep alive a tiny little baby that is 300 grams. And if you want to know how tiny that is, go down to your kitchen, find some scales, and then weigh out 300 grams of sugar or rice or something, and feel just how small that is. It really is phenomenal. So back to the biology A-level course then. Directional selection is our next type of selection, and this is what we're going to see if we get an environmental change occurring. So when we get this environmental change occurring, we get a new selection pressure, and as a result of this new selection pressure, a different phenotype may now be the advantageous one. So if we've got a different phenotype being more advantageous, those organisms that demonstrate it will be more likely to survive, and therefore they will pass on that allele. Which means over many generations, we're going to see this new allele being passed on because it is now the advantageous allele, and as a result the frequency of it will increase. So what we see is this little graph here. Now you can see we've got a little dashed line that is actually our original population, and then we've seen a change in our environment which has led to obviously this new allele being more advantageous because it's conferring a phenotype which has given that advantage in this new environment that we find ourselves. As a result, we no longer have the majority of the population with this allele. Instead, we've shifted it to here. So the directional selection, you're going to see this sliding of our curve, either left or right, depending on obviously what the change is and whether it is obviously an increase or a decrease in that particular phenotype that is going to be beneficial, and therefore the curve moves. So when we're selecting for this new phenotype, remember, it's not an immediate change that we see in the whole population. We are going to see this allele frequency changing over time. So it's directional selection, new selection pressure, different phenotype is advantageous, leads to other organisms then surviving that have that phenotype, and over many generations of breeding, that allele type is going to then increase in frequency. And what we tend to find is that once we've had that directional selection occur, we tend to then get a stabilizing selection period. And obviously that will stay in place as long as this environment remains unchanged. But if we get another environmental change, then back to the directional selection. So what we can see is this kind of shifting between the directional and the stabilizing selection, depending on what's happening in the environment. The next thing we need to consider as far as factors that affect the evolution of a species then is this concept of genetic drift. Now, what we're looking at here is if we've got a population that's descended from only a small number of parents, we will have limited variation within that population. Now, if we end up with a scenario where something occurs that leads to the death of many of those population, we could actually lose alleles altogether. So if we've only got obviously a small number of organisms within a total population, they've all descended from a small number of parents, then one catastrophic event that kills off a significant number of our population there means we could absolutely lose those alleles for good from the population. Now, clearly we've still got some organisms left over. They haven't all died, bless them, but those small number of individuals that are left over, they're going to breed together, but when they breed together, we don't have the same number of alleles within our gene pool. So what we can actually see is that as their population size increases, there's going to be less genetic diversity than there was originally. And that's all down to the fact that some of those alleles were lost due to whatever catastrophe befell them and wiped out a number of their population. That means we've got a much smaller gene pool, much less genetic diversity within that population. So that's what we mean by our genetic drift. And when we think about what causes genetic drift, we could have this as either genetic bottleneck or as the result of the founder effect. So when we're talking about the genetic bottleneck then, this is where we do have that sharp reduction in the size of a population as the result of some kind of environmental catastrophe. So think things like a volcanic eruption or something that wipes out huge numbers of our population, just like that. So at that point, we've reduced the genetic diversity, and then when it expands again, lower genetic diversity exists within the whole population. So what we can see here, not only that smaller gene pool because we've lost some alleles, but we may also see other problems coming out after this genetic bottleneck, because in that population that remains, if we had some of these harmful alleles present, and originally they were in relatively small numbers, now they could obviously increase in frequency disproportionately. So what we could see is a much larger incidence of these harmful alleles present within our population that could very easily affect the overall survival chance of that population of an organism. There are some instances though that we have an improvement in the gene pool. And what we're seeing here is if there was an advantageous allele that happened to be present in our survivors of our catastrophic event, then obviously as the population increases in size again afterwards, they're going to have this advantageous allele. So what we can actually see is our population could actually all end up with that advantageous allele being present. So it can be minor benefit, but all too often not so much, because we've lost those alleles and therefore that genetic diversity is reduced, which just makes them more susceptible to other changes. So the last thing we're going to look at in this video is the founder effect. And what we're going to look at here as our example are the Amish community of North America. So when we talk about this founder effect, what we're really looking at is where we've got a small sample of an original population that then establishes itself in a new area. So what we're going to see is, as a result of this little small group basically moving themselves to a new area to establish themselves as a new population, they don't have such a diverse gene pool as the original parent population did, because there's only a small number of them, therefore smaller number of alleles are going with them. So the reason we use the Amish as a good example of this is because the Amish actually originated in Europe, so around Switzerland, etc. And what we then find is in the 1600s, then we start to see this initial movement of folks from Europe over to America. And this is where we get our Pennsylvania Amish first being established. And in fact, it started off with just 13 families. So if you imagine at that point, we've taken from the much wider Amish community that exists obviously within Europe, and then we have now just taken 13 families across the ocean and placed them in this new environment. Now, if you know anything about the Amish community, then you know that they are really quite insular. So they believe obviously in keeping themselves to themselves. They don't really believe in marrying outside of the Amish community. And so from those 13 families, we obviously would have had a lot of people that would have married amongst the 13 families. Now, that does mean we've got quite a small gene pool in that population that's now established in Pennsylvania. And when we do have this very small gene pool to play with, what we are going to see is that potential for there to be these harmful alleles that in the larger population, not a big deal. But when we're only talking about 13 families that are then obviously going to give rise to all these other generations after them, what we're going to end up with is an accumulation of those harmful alleles. And the reason that the Amish, in particularly the Pennsylvania Dutch, are very useful to use for this is that they actually demonstrate a disproportionate amount of conditions that are caused by these recessive alleles. So things like dwarfism, polydactyly and things like this, that ordinarily you wouldn't really see because they're recessive, so therefore you'd have to inherit both of those recessive alleles to demonstrate the feature. But here, because obviously we've only got a very small gene pool to play with, then they're more likely to inherit those recessive alleles. And as a result, we see much greater quantities of those particular conditions than we would do in a wider population. So founder effect is all about this idea of having a much smaller sample of an original population that then has this more limited gene pool. And that does mean there is a higher probability of them having these potentially harmful alleles in higher frequencies than they should. As always, I hope you found this video useful and interesting, and of course don't forget to subscribe to the channel so you can see when more videos are uploaded, and of course head on over to my website where you can find a range of other resources to help you in your A-level biology studies.